So welcome to Biomindscape. Today we are going to know about the chemotaxis in bacteria. So chemotaxis is a movement of an organism like bacteria in response to the chemical stimulus. It can be positive, it can be negative. The positive chemotaxis is that it moves towards a chemical attractant like nutrient. The negative chemotaxis is the movement away from an harmful substance like toxins. How does bacteria move? A bacteria like E. coli move using the flagella, a long pipe-like structure, and they use the two type of movement, the run and the tumble. So the run is a straight line movement, flagella rotate counterclockwise, and the tumble is random direction change where the flagella rotate clockwise, and they alter between the run and the tumble to find a favorable environment. How does this bacteria sense a chemical? A bacteria doesn't have brains, but they use chemoreceptor on the surface. These are the chemoreceptors. They are known as the methyl accepting chemoreceptor protein. So they are MCPs, and these MCP detect the attractant or the repellent and send signal inside the cell. Now signal transduction pathway in chemoreceptor are MCPs, CheA, W, Y and Z. So these MCPs detect a chemical gradient. CheA is a histidine kinase that autophosphorylates. Then CheY is linked to MCPs to CheA, and HEY receptor get phosphorylated by HEA approximately P and interact with the flagella motor. Then CheZ is it removes the phosphate from the CheY to reset the system. Then R and B, these are the adaptation enzymes that methylate or demethylate the MCPs. And if attractant increases, then CheA activity decreases and less YP receptor are made and less tumbling is done and long runs. So if repellent increases, this CheA activation increases and more CheYP will cause more tumbling and the direction changes. Then adaptation mechanism, the bacteria remember past chemical level using methylation of MCPs by the R and B. So this helps them comparing the current and previous environment, whatever occurred with this chemotaxis is the movement towards and away from the chemicals, and flagella help in the run and turn motion. MCP detect the signal and passes through the CheA kinase. Then they pass to the Y1 and then motor rotation. The adaptations Y R is methylation and B is demethylation. So bacteria doesn't move randomly. They bias. They move based on the chemical clues. So what do we learn in this video? The chemotaxis is how a bacteria move toward and away from the central chemical. Chemo means chemical and taxis means movement. So basically chemotaxis is a way bacteria smell things in the environment and swim accordingly. They move toward the good things like food called the attractant, and they move away from a harmful thing like the toxin called repellents. So how do bacteria move? Bacteria like E. coli have flagella, and these tiny tails that spins and help them move, and they have two types, the run that is moving straight and tumble that is stop and spin around a random. The flagella spin the other way, and running is the flagella spinning in only one direction like a boat engine. Now how do they know where to go? They have a special chemoreceptor called methyl accepting chemotaxis protein. They are present on their surface and these sense the chemical outside. If they are more attractant, they tumble less and keep swimming the way. And if there is a repellent, they tumble more and change the direction and escape. They don't have an eye or brain, but they can measure if the things are getting better or worse chemically. So the signaling system, instead of bacteria, there are tiny signal system with a few main protein. MCP, they are the receptor which sends the chemical. Then CheA, it is a kinase, it starts the signaling inside. CheY, it tells the flagella to tumble. CheZ turns the CheY off. to stop the tumbling, and then CheR and or the CheB adjust how sensitivity the receptor are, so they don't overreact. When something goes in sense, the system reducing tumble, bacteria swim more directional toward it. When conditions stop improving, the system increases tumbling, bacteria try new direction, and this is called temporal sensing. So temporal sensing, and they compare the past and present to decide which way to go. Then adaptation is how they don't get confused. Over time, bacteria adapt to a constant presence of chemical. They stop responding unless a chemical concentration changes, and this happens through methylation. They add methyl group to MCPs to adjust their sensitivity. There is a receptor clustering. The chemoreceptor group together and the member, especially at the poles of the bacteria, and this amplify the signaling so they can respond to a very tiny chemical changes. So bacteria move using the run and temp. They sense the chemical using special protein. They swim towards the food and away from the danger. They remember and adapt by the modifying their receptors, and they are sensitive even though they are so small. So the signal transduction pathway inside the cell, when the MCP sends a chemical, they send signal to the protein inside the bacteria. So we know the names of the important protein. There are CheA is a kinase that start the signaling by adding the phosphate to other protein. CheY, when activated, phosphorylation. It tells the flagella to tumble. CheZ deactivate the CheY so the bacteria can go back to the running. And CheR and CheB help in adapting to a constant signal by modifying the receptor with methyl group. What happens in steps? MCP sends the chemical. If it is good, it reduces the CheA activity, and then it moves forward to the CheY, and it is not activated, and less tumbling is seen and more running is seen. So if it's a bad or no change is seen, then CheA stay active, and it then follows with the CheY getting activated and more tumbling is seen. So adaptation, why bacteria don't get stuck. If a chemical is always present, the bacteria slowly adapt to and stop the reaction. So they do this through methylation of the MCPs. So the CheR add methyl group, making the receptor less sensitive, and CheB remove the methyl group to make the receptor more sensitive. This means the bacteria are always turning their chemical nose. They only respond when things changes. Then receptor clustering, when the MCPs, the chemoreceptor, cluster together at a poles of the bacterial cell, and this grouping make them very sensitive. So every tiny change in chemical level can detect, and these clusters work together, kind of like the teamwork, to decide how a bacteria should move. Then it has some evolutionary importance. The chemotaxis is not only found in bacteria, it also happens in immune cells like neutrophils, which move toward the infection, sperm moving toward the egg, then certain cancer using chemotaxis like pathway to spread. But in bacteria, it's ancient, highly efficient, and survival strategy. Simple example of E. coli. When an E. coli is dropped in sugar water, it sends the sugar using MCPs. It reduces the tumbling and move in the direction of more sugar. If the sugar stop increasing, it tumbles and tries a new path, and it adjusts sensitivity over time to stay responsive. Now let's know about the chemotaxis in immune cell. Cell involved. Mainly neutrophil, monocyte, macrophages, dendritic cell and T cells are involved. What happens here? When there is an infection or injury or a damage to a tissue, a pathogen releases a chemical signaling called the chemokines, cytokinin or a damage signaling, and this chemical signaling form a gradient of higher concentration near the infection to the lower away from it. So immune cell sends their grading using special surface receptor like G protein couple receptor, which we will know about GPCR. So many immune cells can detect the direction and intensity of extracellular chemical gradient and migrate toward the source of stimulus, and this process is called chemotaxis. It is essential in immune system function and homeostasis, and its dysregulation is associated with serious diseases. So I have told about the G protein couple receptor. The chemotaxis is initiated by chemoattractant that bind to the heteromeric G protein couple receptor. which translate the gradient into accurate directional migration. A necessary step in the process is called cell polarization. So, cell migration is important in biological events, including the embryo implantation, developmental patterns, wound healing, and in immune system also. It is important in migrating. A prominent component of leukocyte mediated removal of invaded microorganism and infectious cells from that organism. Chemokines and the receptor regulate leukocyte migration in response to inflammation, as well as homing of dendrite cells and native lymphocytes to secondary lymphoid organ, which enable adaptive immune response and tissue homeostasis. So, as a corollary to the role of the cell migration is established as a function of immune system deregulation movement of the immune cell, and is associated with the development of many pathological conditions, including the autoimmunity, chronic inflammation, immunodeficiency, and cancer. So, chemotaxis role in immunity in immune cells, it can migrate in three ways. Chemotaxis refer to the cell ability to move toward the source of soluble chemo-attracting molecule. So, the immune cell can migrate in three ways. First, the chemotaxis refer to the cell's ability and move toward the source of a soluble chemo-attractant molecule. Then, chemokines is the increase in random unit undirection, the cell motility, and hepatotaxis is a cell migration towards the gradient of an immobilized substance. So, whether these processes are governed by the same molecular mechanism is not known, but three basic principles can be applied to all of them. The first is the cell must become polarized to migrate, and this means the migrating cells and morphologically and functionally asymmetrical with two opposite components. So the leading edge at the front and the unroped at the rear. So second, migration is a cyclic process involving an extension of pseudopodia, philopodia, or lemopodia. So at the cell front and retraction of at the cell back. So finally, the ability to move in a balance by cell adhesion to extracellular matrix, that is ECM, at least is in vitro. There are some sticky surface cell flattened but cannot crawl, whereas on insufficient sticky surface cells cannot generate force to move forward. So our present understanding of cell migration is derived from the studies of different cell type, modes of migration, and environment. This variation explain the studies about the process. So some components are nonetheless share under different conditions, indicating solid element in the cell migration program. So chemoattractant receptor and sensing devices are important. Chemoattractant are the spital signals that initiate and maintain the cell polarity during chemotaxis. Most known chemoattractant bind to the 7 transmembrane receptor coupled to heterotrimeric G protein that contain the G alpha subunit and mediated the chemotaxis through G beta gamma subunit releasing. So, GPCR also governs cell polarity and chemotaxis response to different non-GPCR agonistic. So, this highlights the importance of the GPCR signaling. Then it is also important in P3K, that is the phosphatidylinosol 3 kinase, in amplifying the signaling. So PI3K is a key playing in the grading sensing cellular polarization on the basis of GFP targeted pH, that is the Plex-Stain homology domain, as a bioprobe to detect the spital distribution of PI3K product. So it is important in studying the cytokinin. Then Rho GTPase and their actin cytokinin directs and supports the cell polarity. It increased evidence that in Both the polarity and migration depend largely on the F-actin polymerization of a cell leading edges. Immediately after chemotactic stimulation, motile cell extend an actin-dependent pseudopode at the cell front. According to that, actin and actin binding protein include alpha-actinin, taline, and various things. So, these are related protein ARP 2 by 3 complex located at the front of the migratory cell. Then adhesion receptor are also important as they regulate adhesion strength and critical for enabling cell movement. Adhesion is particularly important for immune cell during the extravasation or intravasation process that takes place during the homing, inflammation, and adaptive immune responses. Regulation of the cell adhesion not only stabilize the nascent lamella and provide the track force that pushes the cell body forward, but also trigger the polarization itself. It moved toward the source of infection. Now, neutrophils, a bacteria invade a tissue, macrophage at the site releasing IL-8, a chemokine, interleukin 8. So the neutrophil in the blood detect the interleukin 8 and migrate toward the site. They kill the pathogen using the enzyme and reactive oxygen species, etc. This movement is fast, directional, and life-saving, part of the inflammatory response. Then signal pathways. Chemokines bind to the receptor on the immune cell, trigger intercellular signaling by the GPCR. It activates the cytoskeleton rearrangement by the actin and cell form pseudopode and crawl towards the higher signal. So chemotaxis in sperm, let's know about it. The purpose is fertilization. Sperm chemotaxis helps sperm cell navigate toward the egg inside a female reproductive tract. The egg and the surrounding cell release specific chemoattractants such as progesterone. In humans and other small peptides or protein, these attractants create a chemical gradient guiding the sperm. So how sperm flow the signal? Sperm have receptors on their membrane. They detect these attractants and change their swimming behavior. Like bacteria, sperm also use run and turn strategies. The flagellum. The flagellum beat differently depending on the signal strength. This ensures only the most responsive and healthy sperm to reach the egg. Then chemotaxis in cancer cell. The metastasis. Some cancer cell hijack chemotaxis to spread called metastasis. When cancer moves to a new part of the body, these cells respond to the chemokines in distant tissue and migrate these using the blood and the lymph system. Example, breast cancer. Cancer cell expands CXCR4, a chemokine receptor. They move toward the CXCL12, a ligand highly expressed in the lungs, liver, and the bones. So, in this cancer, it is often spread metastasized to lung, liver, and the bones. And these chemotactic guiding system is similar to what immune cell use, but cancer cell use it for harmful purpose. So, signaling pathway commonly used is GPCR, Rho family GTPase, and PI3K ATK pathway. Now GPCR, like in the immune cells, Rho family GTPase for the cytoskeletal movement, PI3K and AKT pathway help in the migration and survival. Cancer cell also often ignore, stop, signal and resist apoptosis, that is the cell death, making the chemotactic behavior more aggressive. So chemotaxis is a movement of an organism like bacteria in response to the chemical stimulus. It can be positive, it can be negative. The positive chemotaxis is that it moves towards chemical attractant like nutrient. The negative chemotaxis is the movement away from an harmful substance like toxins. How does bacteria move? A bacteria like E. coli move using the flagella, a long pipe-like structure, and they use the two type of movement, the run and the tumble. So the run is a straight line movement, flagella rotate counterclockwise, and the tumble is random direction change where the flagella rotate clockwise, and they alter between the run and the tumble to find a favorable environment. How does this bacteria sense a chemical? A bacteria doesn't have brains, but they use chemoreceptor. On the chemoreceptors, they are known as the methyl accepting chemoreceptor protein. So they are MCPs, and these MCP detect the attractant or the repellent and send signal inside the cell. Now signal transduction pathway in chemoreceptor are MCPs, CheA, WY, and Z. So these MCPs detect a chemical gradient. CheA is a histidine kinase that autophosphorylates. Then CheU is linked to MCPs to CheA, and HEY receptor get phosphorylated by HEA approximately P and interact with the flagella motor. Then CheZ is it removes the phosphate from the CheY to reset the system. Then R and B, these are the adaptation enzymes that methylate or demethylate the MCPs. And if attractant increases, then CheA activity decreases and less YP receptor are made and less tumbling is done and long runs. So if repellent increases, this CheA activation increases and more CheYP will cause more tumbling and the direction changes. Then adaptation mechanism, the bacteria remember past chemical level using methylation of MCPs by the R and B. So this helped them comparing the current and previous environment, whatever occurred with these chemotaxis is the movement towards and away from the chemicals, and flagella help in the run and turn motion. MCP detect the signal and passes through the CheA kinase, then they pass to the Y1 and then motor rotation. The adaptations Y R is methylation and B is demethylation. So bacteria doesn't move randomly. They bias, they move based on the chemical clues. Thanks for watching. Do like, share and subscribe.